Yes, ma'am.
Okay,
Thank
okay. So, good evening, everybody. I'm Pooja Mandal, and I'm in fourth year PhD now. So, basically, I used to join these classes just to listen to what Prem Kumar is teaching and just to get some, just to recollect whatever we have read. So, it's very generous of him to love, and also, like, I thought, okay, I'll take
you. No,
one class.
actually, thank you, sir. Okay, thank
So I had thought to take other classes also, but there is because of time and because we have put in a lot of effort for this. So what Prem Kumar is doing is really a great job, and those who are attending live, I think, will be benefited more rather than the listening to recorded classes. So, however, okay. So we'll start. So today's topic that I'll be sharing is gene regulation. I think eukaryotes is over. So gene regulation in prokaryotes. So basically, gene regulation. Why we need gene regulation? If we start from that topic, if we think about it, why we need gene regulation, especially in prokaryotic organisms, what happens is since it is a single cell or single cellular organism, they don't like they only need to produce the enzymes which is necessary at that time, in that particular condition, if they are in certain environment, or if they are exposed to, like, if they are having getting some amount of substrate or something, if they are exposed to a particular environment or particular condition, in that condition they have to uh, produce the particular enzyme and manage the condition or manage that environment. So it is not uh, like it is not necessary for them to continuously produce or continuously uh, it is not necessary for some genes to be continuously active. Even in eukaryotes also it is the same case. So we call such genes as constitutive genes, right, which are continuously active. And there are some genes which are like which need to be regulated, which means it's not necessary for them to be always be encoded. So basically, for the final product of a gene is an enzyme. So what are the factors which affect enzyme synthesis? So it can be either degradative process, or it can be in a biosynthetic pathway. So when it is a degradative process, that is when the substrate is more, we need more enzyme to be synthesized. So that is basic. And in a biosynthetic pathway, when the when we are after the biosynthetic pathway is over, when we get more of end product, we need less enzyme synthesis, which means like it is kind of a feedback inhibition, the one which is in the biosynthetic pathway. So coming to gene regulation, so we all have learned about lac-operon. I think that is the basic thing which we have all studied since, I think, since school days. Since I think if all might have taken biology in 12th standard, so all have gone through lac-operon since school days itself. So when we talk about gene regulation, the first thing that comes to our mind is lac-operon. Okay, so basically there are different types of gene regulation. There is negative regulation and positive regulation. So in this first slide itself, I have given that is what is negative and positive, and further also we'll be discussing about this. So negative means that will have the gene expression will be depressed, which means the genes which are there, which are they are already like they are always expressed, and they have to be repressed or they have to be stopped, like their expression has to be stopped. And in case of positive, like the gene is not being expressed and it has to be activated, it has to be started. So that is the meaning of negative and positive regulation. And this concept of gene regulation in prokaryotes is very interesting actually. And if anybody is understanding the concept, then it's very easy, or otherwise it becomes very much complicated, very much confusing. So nowadays they are not giving, since they are not asking direct questions in SRF exam, so it is very necessary to understand the concept clearly. Okay, so there are different levels of gene regulation. So coming to levels of gene regulation, regulation can happen at the stage of replication. So what are all the stages of in the central dogma? of DNA, that is replication, transcription, and translation. So in that we have different at different stages the regulation can take place, either in the replication stage or in the transcription stage or in the post transcription stage, then translation or post translation. So the example of lac operon which we talk about, that is the regulation is at the transcriptional stage, and post translation, that is feedback inhibition. Feedback inhibition is nothing but in the biosynthetic pathway, when the end product is formed, that end product will be binding to some other enzyme to stop that synthesis of further synthesis of that particular end product. So that is feedback inhibition. So that is post translation stage because it deals with proteins. So after the protein is formed, that protein itself will act as an inhibitor. So this you have to remember that lac-operon is the level of regulation of lac-operon is at the transcription stage. Okay. So coming to operon, operon is nothing but it is nothing but it is like functionally related genes. They are clustered together and in a single transcriptional unit. Okay. So we know that prokaryotes are polycystonic, which means all the different genes are there together, they are transcribed, and when they are together and when they are clustered together in a single unit, it is termed as operon. So this single unit may contain different genes, like different structural genes, then it may contain a promoter for these structural genes, and then an operator in which the regulator genes bind. So we'll talk about what is this structural gene promoter. So promoter, and structural genes, and what is this operator and regulator. So when we talk if you see this question here, what is the difference between structural gene and a regulator gene? So if anybody is interested, they can give the answer, because basically it is there in the name itself.
Yes, ma'am. Structural gene produces the gene product itself, and regulator gene regulates the structural genes.
Yeah, so here the answer will be D, right? The structural gene will encode the protein and regulator gene will control the transcription of the structural gene. Okay.
Yes,
So
ma'am.
structural gene itself is not having a control over its own transcription. Basically, that is what is happening in an operon. So even though structural genes are present, it is being controlled by some other gene, that is the regulator gene. Okay. So coming to the model of the operon. So if we talk about the regulator gene, it will control the, as I said, it will control the transcription of the structural genes. So a regulator gene, it is not a part of the operon. So it is some other gene in some other part of the genome, and it is not directly connected to the operon. So when we talk about the operon, or if a question comes whether a regulator gene is part of the operon, then no, it is not part of the operon, but it controls the operon. Okay. And and also another thing is this regulator protein, or the protein which is formed here. And the regulator protein final endpoint to the operon. That is
Hello,
how it operon. Or hello,
ma'am. Your voice was breaking.
hello.
Yes, ma'am.
Am I audible now?
Yes, ma'am. Yes, ma'am.
Yes, ma'am, it's audible. Can continue, ma'am.
Okay. So the end product of the regulator gene, that is the regulator protein, either by binding to the operon or by not binding to the operon or part of the operon, where the part of the operon where the regulator protein will bind, that is the operator region. Okay. In the operon, we have a promoter, then we have an operator, then we have the structural genes. Okay. The part where the regulator protein from the regulator gene it comes and it binds. So it might bind or it might not bind. So it depends on what kind of regulation it is. So if it binds or if it is not binding, that is how based on the binding, it will affect the transcription of the operon. Okay. So this is the basic model of an operon.
Ma'am, there is
coming to,
little bit confusion in that diagram, ma'am. It
yeah,
is mentioned
yeah,
that
yeah.
operon is a complete structural genes, promoter and an operator. But
Yeah.
that point, that regulator gene is not considered part of the operon. The two are little bit contradictory, like that.
See, regulator gene is some other part. Okay, it is in some other part of the chromosome. Okay, but the product which is formed from that gene, that is the regulator protein, that will go and bind to this operon. This operon is not having a regulator gene in its sequence as such. What are the parts of an operon? If the question is what are the parts of an operon, then there is a promoter, operator, and the structural genes. That's it. That when we talk about the operon, we don't talk about the regulator gene because it is not directly part of the operon. It is in some other part, but that product is affecting this particular transcription of this operon. Is it clear now?
Yes, ma'am. Yes, ma'am.
See, in this picture also, both these things, like this one and this one, these two are not connected. They have given it separately. So this one is the operon. The bracket here they have given. So from the promoter till like encompassing all the structural genes and the operator, it will be the operon. Okay.
Okay, ma'am. I confused because of this promoter and regulator, ma'am. That one, in that separately they have given, na?
Yeah.
The regulator's
And the,
promoter, by seeing that I confused.
okay, understood. So basically, since it is a different part in a different part and it is a different gene, it is requiring a separate promoter. It will not be based on the promoter of the operon. This
Yes,
is
ma'am.
different
Understood.
gene. This is a gene, let's say this is gene A and gene B. It's kind of like that. Okay.
Okay.
So, so coming to the control. So this one is very important, which you have to understand. So once you understand this, then there will be no more confusion. So coming to negative control and positive control, which I already discussed. So it is nothing but negative control. We have to inhibit the transcription. So it is already taking place and transcription has to be inhibited. That is negative control. And in positive control, transcription is not taking place. And when there is a positive control, finally transcription will take place or transcription will be stimulated. That is positive control. Okay. So these two things you have understood. Now coming to inducible. Okay. Inducible is nothing but transcription is off and it has to be turned on. Means it has to be turned on. It will be turned on when there is an inducer. Okay. And in case of repressible, transcription is normally on and it has to be turned off. So this much if you have understood, now we will combine this. So now when we combine this, coming to negative inducible. Okay, negative means what? Transcription is
Off, ma'am.
transcription is, wait, let me tell you. See, when it is negative means we have to turn off the transcription. Understood? Already
Okay.
transcription is taking place. And in normal condition, we have to turn off, like the transcription will be turned off in normal condition. So in the negative case, when there is no inducer, here in the first case, there is no inducer. Okay. So the repressor protein, which is being formed from the regulator gene, it will go and bind to the operator and transcription will be off. That is in normal conditions. And when the inducer is present, let's say inducer is present. So in that case, inducer will do what? Inducer will bind to the repressor. And when the inducer will bind to the repressor, transcription will not take place and transcription will be on. Okay. So in case of negative inducible, which means transcription is, in case of negative inducible, when the inducer is present, okay, transcription will be taking place. That means the repressor which is there, it will become inactive. Okay, and in normal conditions, there is the repressor. So just remember like this. Okay, in case of negative, okay, we have a repressor. Repressor is nothing but a gene which will stop the transcription. In case of negative, the repressor which will be formed is active. It is in active condition. And when it will become inactive, only when we have an inducer. So when is negative inducible, repressor will become inactive and transcription will be on. Now let's see negative repressible. In case of negative repressible, what is happening? The repressor which is present, it will be inactive. Okay. And it will go and bind, since it is inactive, it will not bind to the operator of the operon. And transcription will be on. And in case of negative repressible, which means since transcription is on, we have to turn it off. So the product or the repressor, like some other product, we don't call it as inducer, we call it as activator, or we usually call it as activator. So what it will do is it will make the inactive repressor into active. And then the transcription will be switched off. Inducible means we are inducing it. So finally it will be on. Here also it is inducible. So finally it is on. So when substrate is there, it will be on. Inducible means final result is that whenever the inducer or whenever the substrate is present, the transcription will become on. And when it is repressible, let's say even if it is positive or negative in both the conditions, when it is repressible, the product or the substrate or the whatever the activator or whatever is there, the substrate which has to be further it has to be like the gene has to act on the substrate. So that substrate further finally what we get is we'll get an off condition. So you just remember like this. The repressible, if the word term is there, the transcription will be off in the final condition, like in the presence of the substrate. And if it is an inducible operon or inducible regulation, so in that case, the substrate or the inducer will make the transcription on. So coming to positive and negative is like this. Okay, if it is positive, then the repressor or the repressor protein is in the active condition. So see here, see the A picture and D picture, you can see it is an active state. The regulator protein which is there, that is an active state. And in B and C, you can see that since it is negative, both the proteins are inactive. It has to be activated. So this is how you can just remember it, because we don't get this much time in the exam, right? To understand the concept and give the answer. Sometimes if we don't get time, you can just go on this basis. Okay, first you understand the concept, then you make some tricks to remember. So coming to lac operon.
Ma'am,
Yeah.
is that trick inducible means the gene product will turn on, the transcription will turn on?
Yeah.
And the repressible means the the transcription will turn off.
Yeah. And positive means the regulator protein will be active. Okay. And negative means regulator protein will be inactive.
Okay.
Okay. And okay, coming to lac operon, so we all like know all about this, but still we sometimes make mistakes. How? That is like, what are the things that you need to remember here? One is the scientist name, that is Jacob and Monod, and the year, that is 1961. They have described the lac operon or operon model in E. coli. That is the organism on which they worked, and it is based on the genetic control of lactose metabolism. Okay, so there are 3 genes. So sometimes we get confused what is the order of the genes. It is ZYA. So we know there are 3 genes, that is ZYA, but this is the order. So order is also important. That is ZYA. First gene, that is Z gene. So what the Z gene encodes? It encodes beta galactosidase. And Y gene, and which gene encodes what product? That is also important. Z gene encodes beta galactosidase, Y gene encodes permease, A gene encodes thiogalactoside transacetylase. So these are the three proteins or enzymes which are being synthesized by the three genes. So the first one, beta galactosidase, what it does, what the function of it is, how it breaks down lactose. It converts lactose into another compound, that is allolactose. Okay. Further, this beta galactosidase, what it does, it breaks down along with lactose and allolactose. It forms galactose and glucose. So it is breaking down the substrate into smaller products. Basically, it is like a digestive enzyme. Okay, it is breaking down the bigger substrate into smaller parts. And coming to permease, so what permease does is it transports lactose into the cell. So if lactose is available in the environment of the E. coli, it has to come inside the cell. So for coming inside the cell, there is this enzyme permease, which will transport lactose into the cell. So we all have, like, we might have this doubt. So if this operon is not functional, like when lactose is not there, it does not function, right? So when lactose is there, if it is not functioning, how lactose will come inside the cell? Like if permease is not being produced, how lactose will come? Like at the first, the answer for that is, like, since even though it is off, some amount of enzymes will be. Hello. Yeah. Am I
So,
audible?
Yes,
yes,
ma'am,
ma'am.
it's audible.
Yeah. So even though if it is like not operating at the initial stage, some amount of enzymes will be there in the as a residual amount in the cells, which will allow the initial lactose to transport inside the cell. So after that, once it gets little bit, then further the permease will act and it will actively transport lactose into the cell. Okay. And the another thing which is important is that lac operon is negative inducible. Okay. So negative means what? Negative means the
Repressor will
repressor
be
protein
active
is
stage.
inactive. It has to be, yeah. And it has to be, yeah. Sorry, repressor will be in the active state. And inducible means what? Final product
Final
transcription
product
will be on.
on.
Okay. So here what happens? The regulator gene which is there, it produced as a repressor. So that we'll see in the picture. So in the absence of lactose, what happens, since it is a, here the inducer which is there, which is what the substance which we call as the inducer is nothing but lactose. Okay. So in lacoperon, in the absence of the inducer or in the absence of lactose, what will happen? The normal condition, it is, since it is a negative control, the regulator gene, or the protein or the product whichever is formed by the regulator gene, the repressor, it is in the active state. Okay. So in the absence of lactose, since it is an active state, an inducer is also not there. In that case, it will go and bind to the operator. And since it is binding to the operator, it will not, like when the promoter is there, it will not allow the transcription. So none of the structural genes will get transcribed and translated. But in the presence of lactose, what happens? The active regulator protein is formed. But since the lactose is there, it will go and bind to the repressor. Once it is binding, it cannot further bind to the operator. Okay. So since it cannot bind to the operator, now in this case, there will be normal transcription and translation and all the enzymes will be produced. Okay. And some of the yeah, here another thing which had, like, I had seen this question, I don't know, I don't remember whether exactly in SRF or some other competitive exam, they had asked, like, we know all about lack of brown, but they asked this question about what kind of motive is the repressor. So basically, it is a helix turn helix motif. Okay. So this active regulator protein which is formed here, it is a helix turn helix motif and it goes and binds to the operator. So this also sometimes they ask such questions. So even after you attend classes and all, Like you have to read the books because some points you might remember from the books. So that sometimes it might click in exams. So after this, yeah. So till here it is clear. Now we'll talk about there are some mutations in the lac opera. So if we talk about the mutations, so there are different locus, right? First we can say that there is I locus here, lac I. This is one locus. Okay, then there is lac operator, that is O locus. Okay, and the structural genes are there. Each one is a different locus. So if we talk about mutations, each of these locus can get mutated. Okay, and how they studied this mutation is like, since you see, like bacteria has how many chromosomes?
One single circular chromosome.
Yeah, it has one single circular chromosome, right? So for studying these mutations, Jacob and Monod, what they did is, uh, what they did is they created a plasmid with different mutations, okay, of this particular gene. Like for example, if they wanted to study the operator locus, they created like they had like whatever mutant they found in from other bacteria, they made a plasmid in which they had that particular mutation, and then in a single bacterial cell they had this plasmid along with its own chromosome. So it was kind of, let's say, partial diploid or merozygote. They say merozygote, right? Like, which means that not all of the genes are in diploid condition, but particular, like some genes are in diploid and other genes are in monoploid condition. So they studied in that way, because otherwise it is not like possible to study with single allele, right? If we have to study mutation, we need two alleles. So know how the nature of the mutation and how it is acting and all. So let's say the first one, that is the operator locus. In that, there is a mutation that is called OC, okay, which means operator constitutive. So this mutation in the operator locus, what it does is, what is the function of operator? That is a place where the repressor will bind, right? But if the operator locus itself gets mutated, the repressor cannot bind there because that sequence is mutated, and it is like there's some difference there, because of which the repressor will not go and bind. So this OC mutant, like allele, it is actually dominant over the normal one. The normal ones are usually represented by the plus symbol. And what happens in all of these OC mutants, whether they are in homozygote condition or heterozygote condition, since it is dominant, whether or not, even if lactose is present or not present, irrespective of that, it will be always transcribed. Like all the genes, like the structural genes, will be always transcribed. So if you see the picture here, see, here you are having lac OC, okay, and another chromosome you are having lac O plus. But since this is a dominant, or yeah, this is a dominant mutation. So what happens, since even though it is in the heterozygote condition, transcription and translation will take place, and the gene like this beta galactosidase is formed. Okay. So even in the presence of lactose also, it is the same result. Even in the absence of lactose also, it is the same result. So irrespective of presence and absence of lactose, these genes will be synthesized, and like genes will be encoded and the enzymes will be synthesized. So that is why this mutation is also called as constitutive mutation. And see, usually if such questions come like based on lac operon, usually they give in this form, like this is how they write different mutations. So lac I plus, which means the regulator gene, or this gene, okay, the regulator gene is in normal condition. Plus means normal, so it is a normal condition. Here O plus and OC, you see lac O plus and lac OC, it means that it is in heterozygote condition. But since this is dominant, it will be constitutive expression. And Z plus and Z minus. Okay, Z, like all of the structural genes are also dominant, the mutations which are there, they are all recessive mutations, which means even like if it is in heteros, if one of the chromosome is in one of the chromosome it is in Z minus also, it will form the product. Okay. Now coming to the next locus, that is lac I locus. In lac I locus also, in lac I locus is also like it will result in constitutive expression, which means that I locus is nothing but the locus which encodes the repressor protein. So if there is a mutation in that particular gene, then it cannot produce a functional repressor. Functional repressor will not be produced, and the repressor which is produced, it will not be able to bind to the operator. But in this case, what you have to remember is the normal condition, that is repressor being produced in the active form, that is dominant over the mutant one. So if there is one lac I minus and one lac I plus, this is dominant. So if it is in heterozygote condition, it will not like, it will be like lac operon will function normally. There will be no difference. Okay. And also another thing you have to remember here is that there is like lac I mutant or lac I locus, it is a trans-acting gene. Okay. Lac I plus is trans-acting. Trans-acting means that like it can regulate the gene
Anywhere in
or
the
it can
genome.
anywhere in the genome, not necessary that has to be connected. But lac O wala is your cis-acting. It has to be there in that particular region only.
Same strand.
Yeah, in the same strand. So that is transacting and cis-acting. So here you can see in the lac I plus operon, like the mutant one, lac I plus is there. Yeah. 1 second here. Okay. You have lac I plus and lac I minus. Okay. Even so, even if lac I plus and lac I minus are there, since it is in like, as I said, lac I plus is dominant, that's normal condition is dominant. So one active repressor is enough for inhibiting the operator. Okay. So like even in presence or absence of lactose is not like it is having an effect here, because see, in the absence of lactose there is nothing that can inhibit this repressor, but in the presence of lactose what happens is like how normally lacoperon will work, that is how it will work in case of heterozygote. But if you say, let's say that this particular combination is like if both are lac I minus, then in that case it will not, like it will be a constitutive expression. Even though lactose is there or not there, it will not matter for this. Okay. So that is another, like this is about lac I plus and minus. And then there is another mutation, that is super repressor, lac IS. Okay. In case of super repressor, whatever you do, like from the name itself, you can understand it is a super repressor. Like whatever you do, whatever, like you give how much ever inducer you want, whatever you do, it will always be in repressed condition. It is non-inducible. Okay, it cannot be inducible by anything. So transcription will always be absent, or it will not take place. Okay, even like regardless of lac OC is a constitutive expressor. Okay, and there are other mutations.
Ma'am, how
This
exactly
IPT,
this
the
super
IPTG is
repressor
one of the
works, ma'am?
super repressor is. I think I'm not given the picture for this. So, super repressor is like like
I think it won't respond to the lactose, ma'am.
Yeah, ma'am. Repressor will be modified such that lactose will not bind to repressor.
it is dominant, what is happening is it is like even though in homozygote condition or heterozygote condition, it will not it will make a functional protein, functional repressor. So whatever condition it is there, if this super repressor is there, the protein will be formed in such a way that it can always bind to the operator unless the operator is having some other mutation.
Okay.
Okay. So that is how, and this IPTG is another inducer, like it is called a gratuitous inducer. I have not given, like I have not written it here. It is called a gratuitous inducer. It is like it will induce, okay. It will induce the transcription of the particular operon, but it will not, like it will not like it will not act like it is not, it is similar to lactose, but it is not exactly lactose, so it will not be broken down by the enzymes, since like straight for the enzymes, right? But in case
Ma'am,
of
your
gratuitous
voice is
inducer, it is
breaking
just
in between.
now it's okay. Hello.
Yes, ma'am.
1 second, I went to somewhere else, I think. Uh, Wait 1 second. I clicked on something. Hmm. Okay. Now my hello. Hello.
Yes, ma'am.
Yes,
It's
Yeah.
audible,
you're
ma'am, now.
audible.
Okay. Okay. So what I was talking, yeah, about IPTG. So it is a gratuitous inducer. Gratuitous inducer means what? That it will not act as a substrate for the enzymes, but it will induce the operon. So in case of whenever there is a super repressor allele, not also like they will they are also of no use basically. Okay, coming to, so these are the three main important mutations which you have to remember: lac I minus, lac OC, and lac IS. So sometimes like they give the combination, different combinations, and you just see gene like one, like see gene by gene, and you just, if you remember which one is dominant and other which one is recessive, which allele is dominant, which allele is recessive, then you can easily figure out whether lactose will be produced or not in the presence and absence of lactose. Sorry, lac operon will be operational or not in the presence or absence. So just see one, like one by one, you go gene by to figure it out. So this, in case of structural genes also mutations may take place. So in, if structural gene as itself is getting mutated, then like we can't do anything, right? If even though it is operational or not, even though operon is working normally or not, if the Z gene or let's say Y gene, okay, because these genes are independent of each other, it is not like one is not affecting the other. And if there is a defect in the Z gene or Y gene, in that case, what will happen? Like, will not be able to get any product. But if there is a defective Z and if there is a functional Y, then one of the enzymes will be produced. If there is a defective Y and a functional Z, then the other, like based on which one is functional, it will produce the enzyme. So it is not dependent on one another. So each gene is independent. So that is what we can see, even operator and regulator and all. So we have to see gene by gene, and based on whether that's dominant or recessive in that particular situation, you can then find out whether it is functional or not. Okay. Now this is about how lack of operon is negative inducible. So it can also be positive inducible. So lack of operon, always we cannot say that it is negative inducible, but it is also having a positive inducible control. So how it can be a positive inducible control? In that, like in positive inducible control, what happens is positive means what? Your repressor protein will be inactive, right? It has to be made active. So if it is inactive and it has to be made active, how it will be in this case, in case of positive inducible? So here basically it is the key player here is CAP, that is nothing but a catabolite activator protein. So this is a protein which binds to a site near the promoter of the LAC genes. Okay, and what the function of this protein is nothing but will enhance the RNA polymerase to bind, and it will enhance the transcription. So that is what this catabolite active cap protein's function is. And the requirement for this cap protein is nothing but it has to form a complex with CAMP, that is cyclic AMP. This is another nucleotide. So CAMP is nothing but it is a modified nucleotide which is involved in cell signaling. And how it regulates is that, so basically you just understand that CAP and CAMP should always be together. Okay, and CAP and CAMP together, they enhance the transcription, or they enhance the RNA polymerase binding, and further they enhance the transcription. But the CAMP, it is regulated in different way. How it is regulated is that it is inversely proportional to the amount of glucose which is present in the cell. Okay, so why here we are talking, suddenly we shifted from lactose to glucose? So that is one big doubt here. Okay, because we are talking about lacoperon. Lacoperon's function is to break down lactose. So suddenly now we are talking about glucose. So why we are talking about glucose is the final product or the smallest unit of lactose, right? After the breakdown of lactose, it forms glucose. Finally, glucose is the product that the cell needs. Okay, so that is why glucose is also very much important for the functioning of lacoperon. How? That we'll see here. So since glucose, the amount of glucose in the cell is inversely proportional to the concentration of CAMP. So if glucose is high, CAMP will be low. And if glucose is low, CAMP will be high. So further what will happen? So in case of high glucose, if you see, there will be low CAMP. And if there is low CAMP, it will form less of CAMP cap complex. So why this complex is needed? For transcription. So if this is low, then transcription also will be low. And in case of low glucose, CAMP concentration will be high. And there will be more of binding and will increase RNA polymerase binding and finally increase transcription. So that is how it is, this positive inducible control takes place. Okay. So if you see this picture, you will understand more better. That is when glucose is low. Okay, glucose is low. That let's say lacoperon is there and it is hungry. It needs food. So its food is glucose. When the food is low, what will happen? Whatever lactose is there, it will break down and create is food. It will be satisfied. But when you are directly giving the food, so it is like if you are not getting food, you go to the mess and eat, right? But if your food is given coming to your room, then you won't go to the mess. So it is a situation similar to that. So when the food is readily available, that is glucose is in the high state. In case of when the glucose is high, why it will take the effort to do all these like transcription, translation and all. So it will just be happy with whatever glucose is there. So this process will not take place. So the logic behind this is that when the glucose is high, CAMP will be low because it is inversely proportional. And CAMP is low means it will not bind, like less binding will take place, and there will be like RNA polymerase and all will not be active, like it will be not enhanced, and then there will be low transcription. So this is what you have to remember. Sometimes questions come in this form, that what happens when glucose is high, lactose is low. Glucose is high, lactose is high. Okay, glucose is low, lactose is high. So different combinations of questions they ask, and what happens? That is what they ask. So this is how you remember it. That is influence of glucose on lacoperon. So when glucose is high, glucose is present and glucose is absent and lactose is present. So in that case, you have to go to the mess to eat. That is, transcription and translation will take place normal on the operon. But in any case, if you see glucose present is there, if glucose is present is there, just don't see what the condition of lactose is. Glucose is present, and if it is in high, then lac operon is repressed, or lac operon will not be functional very much. Okay, so this much you can remember about, like, I hope you have understood about the positive regulation of lac operon. Okay, so for
Yes,
the
ma'am.
next one, yeah, I think this till here it is clear, right? Any doubts you have?
No, ma'am.
Okay.
Ma'am,
So
glucose
now,
actually affect that cyclic AMP, ma'am. Means the once
yeah,
go to the previous slide.
this one,
CAMP.
yeah,
That
cyclic AMP.
those two products, those two substances are there now, ma'am. CAMP
Here,
before
here,
CAMP.
here. The first, this first picture or
Normal
second one?
AMP and CAMP. Normal AMP,
Hmm.
ma'am, that one. Glucose will affect those two binding of those two substances.
No, glucose is affecting the concentration of CAMP, cyclic AMP. Okay.
Okay. It's not
This,
affecting those reaction, ma'am. The reaction
no,
between
it
those
is
two.
not affecting this reaction. This reaction is independent. Okay. This cap is already there in the cell. Like, that is being produced by some other genes and enzymes. That is not being regulated. The regulation is here. Like, glucose is affecting this one. Okay.
Okay.
This CAMP. So if CAMP is more, then more binding will take place. It is not affecting the binding. If there is
Okay.
high, if it is in high, then binding will also be faster or higher. So basically that is what happens. Now, when if there is more of substrate, then there will be more of you. If you have studied in like in biochemistry and all, we study this, what is that called, when we study enzyme regulation and all, no? So they say that there is competitive inhibition and all such kind of things. So what happens is, if one particular substrate is more in concentration, it will be like, it will bind
Hello, ma'am.
more of CMP, then there will be more of binding efficiency. So this will happen only when glucose is not there. And glucose, when it is high, so glucose is affecting the CAMP concentration. Okay. So till here, if it is clear, we'll move on to the next operon. That is nothing but TRP, or tryptophan operon. Okay. Tryptophan operon is discovered by Janowski. So I've not mentioned.
Hello, ma'am.
Example of negative is by the negative, is a negative repressible operon. Okay. So here what happens is, negative means what? Repressor or regulator protein is inactive, right? And repressible means final product will be, translation will be off. Okay. So here what happens is, in case of tryptophan operon, in tryptophan operon, it is little bit, the operon structure, it is little bit different from that of lac operon. Okay. So is it audible?
Ma'am, sometimes it is breaking, ma'am.
Think there's some internet issue.
Yes, ma'am, some network problem.
Uh, okay, just tell whenever it is breaking. Okay, we don't.
Okay, ma'am.
Yeah. So tryptophan operon's model is little bit different from that of lac-operon. In lac-operon, we had only 3 genes. Here we have 5 structural genes. Okay, A, B, C, D, E, but it is in the reverse order, D, E, D, C, B, A. Okay, and all the transcription and translation is so how normally it takes place, and all the enzymes together, it will form the tryptophan. Okay, so it is when tryptophan is low, this operon should function in such a way that it produces tryptophan. And when tryptophan is already there or when tryptophan is high, then no transcription will take place. So how this happens is here, since it is a negative operon, your repressor is normally inactive. Okay, if it is inactive, what will happen? It will not bind to the operator. So operator binding is not taking place. And if the operator binding is not taking place, the normal transcription and translation will occur. Yeah, normal transcription and translation will occur. And in the other situation, when tryptophan is high, in this situation, we have an inactive regulator or repressor. But this tryptophan, it will go and bind to the repressor here and it will make it active. So once it is binding to the repressor, it is making it active. Now this active repressor will bind to the operator and transcription will not take place. Okay. So it is a negative repressible operon. This is how it controls the biosynthesis of tryptophan itself. So we talk in terms of whether tryptophan is there low and high. So here we are not talking about any inducer. We are talking about that particular product of the operon itself.
We can call co-repressor na, ma'am, tryptophan, co-repressor.
We call it as co-repressor. That's what I was saying. Like we don't call it as inducer. So since it is making a inactive repressor into active, so it is called a co-repressor. Okay. So how here the functioning of TRP operon is taking place. So here you can see that there are 4 regions. Okay, 1, 2, 3, 4. 4 regions in the 5' UTR region. Before the structural gene starts. Okay, before even the structural gene starts. Here you can see there is a 5' UTR region. So 5' UTR after which the structural gene starts. So 5' UTR here is very important because the regulation takes place in the 5' UTR of the TRP operon. So how it takes place is that 2 structures form. Okay. So basically this one is different. Like negative repressible operon is, okay, this is one kind of regulation. So there are 2 more kinds of regulation in TRP operon. That is what, like in that 2 kinds only 5' UTR is important. Okay, did you understand? Like if we talk about TRP operon, there is not only one kind of regulation. There are 3 kinds of regulation. Basically, one is negative repressible. That is simple. How we are seeing in this picture. How the tryptophan acts as a co-repressor and all. But in the other kind of regulations where the 5' UTR region, that is the regions within the 5' UTR, that is 1, 2, 3 and 4, they play a major role. How they play role is that they form 2 secondary structures. So one is called as the terminatory structure and the other one is the anti-terminator structure. Okay. So first one, you can see this picture. If the region 1 and
this process is called attenuation.
Yeah, attenuation. So the terminatory structure which is formed, it is called as attenuation. That means that it terminates transcription. Whereas the second one, it is nothing but anti-termination. So how it is formed? It is 1 and 2. In case of terminatory structure, the regions 1 and 2 and regions 3 and 4, they bind. after which we have a string of uracils. Okay. And in case of anti-termination or the secondary structure, in that case, the in this case, one will not bind with anything, and 4 will also remain free. In the middle portion, that is 2 and 3 will bind together and form a hairpin. So these are two kinds of hairpin that are formed, okay, in the 5' region of the mRNA transcript. So how this affects the regulation? So when the tryptophan levels are high, we need termination. We need to stop. Okay. Which means we need attenuation. That is called attenuation. Termination is nothing but attenuation. Okay. We need the process to stop because tryptophan is already high. So in that case, 1 and 2 will form hairpin. 3 and 4 will form second hairpin. And this string of uracil, this is what will helps here, because the string of uracil is acting as a terminator sequence. So once it is acting as a terminator sequence, RNA polymerase gets terminated and transcription of TRP structural genes will not take place. So what's the significance? It is nothing but it will prevent unnecessary synthesis of tryptophan because it is already in high condition. Whereas in the second situation, that is anti-terminator structure, so when tryptophan is low, so in that case, we need tryptophan. The cell needs tryptophan. So in that case, 1 will not bind with anything. 2 and 3 will form one single hairpin. So there is single hairpin formation. And in attenuation, there are 2 hairpin formations. Okay. So when 2 and 3 binds, after this hairpin, there is no string of uracil nucleotides. So basically, when in RNA termination, when we talk about RNA termination, there is usually a hairpin followed by a string of uracils. That is the structure. That is why it will recognize the structure and it will stop in case of it will get terminated. Transcription will get terminated in case of attenuation. But since that structure is not formed in anti-terminator sequence, so your termination will not take place, and it allows the tryptophan enzymes it will form tryptophan. Finally, tryptophan will be formed. Okay. So how and why these structures are formed, I have given little more detail here. So basically, it is not that much required, but still you can understand why in case of when there is high tryptophan, it is forming attenuation is happening, and why in case of low tryptophan, anti-termination is happening. So in this case, you can see when tryptophan level is low. Okay. So when tryptophan is low, what happens is first your RNA polymerase will start transcribing. So region 1 is produced. Okay. In case of bacteria, you have to remember this, that transcription and translation, it takes place simultaneously. So it is not that once transcription takes place, then mRNA will be formed, then only translation, but it takes place simultaneously. Once transcription starts, behind that itself your ribosome will get attached, and simultaneously your translation also takes place. Okay. So when tryptophan is low, what happens? This transcription is started. It is going on. So once it is finished, the region 1, okay, there is tryptophan codons. So this portion, it needs a tryptophan for further synthesis. So since tryptophan is low, what happens is, even though ribosome will be coming here, okay, it will stop here for some time. In this third picture, you can see in the tryptophan codon region, the ribosome will be stalling. It is called stalling of ribosomes. So in this region, it will be stopping for some time. So why it is stopping? Because there are tryptophan codons here. So to get since tryptophan concentration is low in the cell, to get the tRNA charged with tryptophan, it takes time. So it will be stopping here for some time. So since it is stopping here, meanwhile, transcription is taking place. In this region 2, it has got transcribed, region 3 also got transcribed. So since these two regions got transcribed, so by the time this ribosome is coming forward, these two regions will form a hairpin. So that is why region 1 will remain free and region 4 will also remain free. whereas in between portion will form a hairpin loop, and transcription will continue. So attenuator is not formed here. So it is anti-terminatory structure is formed here. Termination will not take place. But when the tryptophan level is high, the case is ulta, or it is different, because here it will not stop for some time. This the third picture, the ribosome will not stop for some time. It will just be moving along with the same pace along with transcription. So since it is moving in the same pace, this 1 and 2 region will form hairpin, because 3 and 4 region will transcription will always already be taken place, and it has already formed a hairpin there. So in this case, you will get a attenuator or attenuation will happen, and it will stop, like transcription will stop. So how you can remember this, that region 1 and 2 gets binded, like 1 and 2 will form hairpin and 3 and 4 will form hairpin. Like how you remember which one is attenuation and which one is anti-termination? Because such kind of confusions will happen only when we give the exam. Like while giving the exam, you get such doubts. So because we know many things, but we get confused. So how you remember is, if we have to stop transcription, if transcription is not taking place, it is like from first itself it is stopped, which means on the first region itself there is a hairpin. So you can remember like that. But if transcription has to take place, then the starting is not like starting is free. You just remember like the first one. Okay, the first one is bound, then transcription will not take place. And the first one is free, then transfer. These are the 2 points which you have to remember here. So when there is high tryptophan levels, attenuator structure forms, and transcription terminates, and no synthesis of TRP enzyme. And when there is low tryptophan levels, anti-terminator, and transcription will continue. So that is it about, I think, this gene regulation in eukaryotes. Is it clear?
Ma'am, in
Yes,
both
yeah.
the cases, that hairpin loop has formed, but the transcription is going on. Translation is going on in one case, and it is not going on another case. Why it is like that, ma'am?
That only I said. Like, uh, see, for translation to stop, okay, like sorry, transcription to stop. For transcription to stop, what happens, what is required, like what is needed is that the terminator sequence, if you have read about transcription in detail, you will remember that the terminators is like the sigma factor will be acting, right? So the sigma factor, what it will do is like it will recognize a particular sequence. That sequence is nothing but one hairpin followed by uracils, followed by string of U. That is the sequence that it is recognized, that usually recognizes that is the region, or that is the terminator sequence ka structure, secondary structure. So based on that structure only, termination or stop, like transcription will stop at that particular point. So even though both cases, the hairpin which is formed, it is followed by this string of uracils. Okay, the hairpin formed followed by string of uracil. So this particular structure, it is acting like a terminator. That is why transcription will stop. But in this case, here the hairpin is formed here. Okay, followed by the hairpin, there is no string of uracil. There is another region. That is nothing but the region 4 of the TRP operon. So after which, okay, U is there. But hairpin followed by U is not there in this second picture. But in the first one, or the attenuation case, hairpin followed by U. Hairpin followed by U is nothing but it is similar, or the terminator recognizes this sequence as a like the portion where the transcription has to stop. So that is why it is like this one is acting like a terminator of transcription, whereas this one is not. Like this one is normal. Like it is not, this is just a normal sequence.
Okay, ma'am.
Okay. So if you have any other doubts in any other region or portion, you can ask. But I think I have covered, like these are the basic things from which questions may come. There are many other things, but you can read some basic books where you can get your concepts clear. And uh, sometimes, like whatever I have discussed, basically they ask from such like, but nowadays they ask reasoning kind of questions. So your concept should be clear and you should not get confused in the exam. So that's why you make some tricks to remember certain things. And I think you can read some books also, because once we understand, later on we forget also. So you have to be in revision continuously. Or any other doubt you can ask.
No, ma'am, no doubt.
Hello, ma'am. I'm audible.
What?
Thank you, ma'am. It was a nice presentation, ma'am. Very informative and like it's very good presentation, ma'am. Thank you, ma'am, for giving your time and efforts, ma'am.
Thank
And
you. Thank you
from
for
day one, supported very well, ma'am. Like your feedbacks helped me a lot. And like, hello, ma'am.
hello.
Yeah, ma'am. Like, like if you want to give some guidance, you can give, ma'am. Like for SRF tips, like students will be helpful. If anything you want
Okay.
to say, you can say, ma'am.
Now I think only three people are there, but
Yeah, no,
I
I'm
think you have.
recording many listen, ma'am, actually. Because I'm
And
getting data, ma'am, actually. Like
mhm.
who will watch the how much recording? Like I will get the data, now, ma'am. Almost
Okay,
all,
okay,
most of them are watching recording, ma'am. Within 2 to 3
okay.
days, everyone will watch. Yeah.
Then it is good. And tips in the sense, I think almost in every class you have given tips. So there's nothing
Mm.
extra that I want to say. Only thing is don't get confused in exam. So for that, you need to do more revision, and possible you try discussing topics with somebody, friend or senior or junior or some person, because through discussion you will get more knowledge and your doubts will be cleared. You'll remember more things simply just by studying alone. Better than that is if you can discuss some things and study. And other thing is that try to read some books also, because in MSc only you find some time to read. After that, you like we don't once we go get into research deeply, we don't read basic books. So we always don't
Yeah, ma'am.
don't look for shortcuts in some objective book or some key notes kind of things. Okay, when time is less you can do that, but mostly try to read some basic books also along with these some uh, whatever are there, like short notes and all, and try to make own notes and read. But I think those who are really interested, they will be doing all these things.
Nice.
And mostly uh, in exam, usually we get confused. So that is one thing you have to take care of. Otherwise, everything is, I think every class you have, Prem Kumar has given some advice. You can follow such all these things, because our subject genetics and plant breeding, we have to have both conceptual knowledge and what's all but ability to memorize things also, because there are many things which we have to by
Yeah,
heart,
ma'am. Well said.
and many things we have to understand. So both are important. So try to manage time accordingly. And now the even SRB net ARS and all has come, so maybe all like your preparation will help in all both all aspects. That's it.
Yes, ma'am. Thank you, ma'am. Thank you, ma'am, for your time, ma'am. And it's
Thank
very
you.
good. Really, and I have to thank participants also, ma'am. Like all the classes they have supported well, and actually, I think
Yeah,
course finished after I don't know how 2 months has passed. It's very really short.
but I
And
really
actually, ma'am, I don't.
congratulate
I,
you because it's not very easy to make slides and
yes,
take
ma'am.
class.
Yeah, ma'am.
Because
Actually,
for one class, I like for this class itself, like I understood. I know also that it is very difficult, and to
yeah, ma'am.
teach such molecular concepts or such kind of things like basically all this molecular genetics and such things to teach in online or such in PPT mode,
Yeah,
it's
ma'am.
really difficult actually.
Yeah, ma'am. Actually,
The
making slides it will take very much time, ma'am. Actually, I used to take
Mhm.
all day to prepare slides, ma'am. Actually,
Yeah, so
somehow
we
I
need
finished,
more.
ma'am. Luckily, yeah. Uh,
Okay, now next time onwards you can improve on whatever slides you have.
yeah, ma'am. Maybe this is will the last maybe batch, ma'am. I think I will not be
Okay,
continuing. Yes, ma'am.
okay. Mm.
Yeah, but it is actually it's benefited me more, ma'am. Actually, because like while doing presentations, I got lot of doubts clarified, ma'am.
Mm. Mm. Yeah.
Thank you, ma'am.
Only
And your
thing,
feedback.
I think if many people joined and more discussion was there, then
Yeah,
it would
ma'am.
have been better.
Very well.
But
Ma'am.
it's okay. Like anyhow, people are listening
Yeah,
to
ma'am.
recordings and asking doubts also. It is okay.
Yeah, ma'am.
Only time is the
They're
problem, I
asking.
think. So, hmm.
Yeah, ma'am. Time because sometimes maybe many will be in hostels or many will be in they
Yeah.
maybe don't have facilities. That's the
Hmm.
problem. Okay, ma'am. Thank you, ma'am. Really, it's
Okay,
a very
then.
good presentation.
All the best.
Yeah, ma'am. Thank you, ma'am.
Thank you very
Bye.
much.
Good night, ma'am. Good night.
Hmm.
Yes, ma'am.
Okay, bye.
And thank you all participants. Really, it's very good journey, and this will be the my last variation. I think like last word. And only thing like before ending the course, like maybe my friend will take another one class mutation. Uh, like after that, maybe we are not taking any classes, and please attend. Please watch recordings, and if you had any doubts, you can ask at any time, and you can text any time. And if I will, at least if I not respond immediately, definitely I will respond. Like I think 1 or 2 days after. And please, and also discuss among you more. Like it's very difficult, as Madam said. Uh, Discussing with your friends is more important actually. Like what I will learn most of through discussion because I have one friend, Big Bon, and we used to learn together. Like when I was learning Linux operating systems and some different analysis, we used to learn together, and we learned more actually in very short duration of time. And my seniors is guided in that way to learn together, and even sharing the information, it's very important. Don't feel like competitive among yourself, among your batchmates. Okay? Always try to like try to discuss more, and uh, give time for yourself also, but at least 1 hour you to try to discuss the topics so that it will get topics get evolved, as Madam said. And it's really worth it actually, because in area we used to do like this, in canteen we used to discuss a lot of things. And so, and the environment also, like if you start doing, the environment also will be supports you. That's how in IRA, like the positive environment, because of the students will share more actually. They will always discussing the topics, research work, and wish to share among us. It will be very, very helpful. In exam time, it will go peak actually. Wherever you see the people are used to discuss, and by looking at them, you'll feel fear and you will start reading. That's what the advantage in our campus. And you should build in your campus also the same environment. And with these words, like if any doubts, and please read, it's very difficult because even getting seat in IRA or even getting seat in higher hubs also is very difficult, because if you are in general, then it's too difficult actually, because hardly how many seats, hardly 10 to 12 seats will be there. And you know how many SRF seats are there? Are only 20 seats. I don't know how they will. Like sometimes they will reduce to 18 also. And also in many campuses, even though you- like CSR will be there, and try to prepare CSR also, and try to prepare UGC. If anything you are feeling comfort, you try to prepare and try to crack it. Okay. And after that, having a fellowship also, choosing a good college is also very important. And you can contact me anytime till the exam is so far on June. Prepare well, and if any doubts in between that, you can ask anytime, even decision making also while choosing the colleges, or while going to attend any exam, other exams also. If you want any guidance, I have so many friends, I can have interaction with you if any time is available. Okay, with this I'm closing my class also, and I'm closing my course also. There will be no live class from my side from today, and most of the topics I covered, I think. No, I covered like what are the topics I given to you, promised, and I have covered all the things. I think like the things maybe sometimes in the books, some detailed will be there in slides, will not be there, because you can't bring a book to a class. Okay, like we can't transfer everything into a class, but the classes should be very informative, like very if you are watching my class, at least you should able to understand the topics in the book, like by that's why I prepared slides in that way. And really, like it also benefited me also, like if I want to say, because while preparing notes also I learned many things, like collecting, gathering information from many books. Like I used to have a books, continue like every book on my table, and I used to read for a one topic. If I choose gene into interaction, I used to prepare from Nandarajan book. I used to prepare from J.R. Sharma book. I used to prepare from B.D. Singh book. What they have given, what extra information is they given, and by compiling that, I used to make PPT, and PPT serve as short notes for me also, and for you also. Okay. And I thought to bring it as a book, but suddenly exam came. I can't do that. Okay. And next month it's not possible. Maybe I think, but I will definitely bring my lecture series as a book, because, but on sharing PPTs also, sometimes last batch I have shared the slides actually. But like uh, some of the students actually they used my slides, and they have used my information, and they published the book actually. I don't know why, but okay, it's happy to be published. But although I'm bringing the book also, and I think it will help for many and help for us also. And if any doubts, you can ask at any time. And I'm saying you for all those who are those who are watching recording, those who are going to watch recording, those are attending live only, two are there. And I don't know why the people are not make the time to attend the live class. Either lectures are boring, or I don't know what is the problem. But somehow the course will end it. But I have given chance also for after joining. Like I given 15 days or 5 days, or 50, I think 15 days time to retake the course. Like. Cancel the admission. But I think that also I didn't get any response. But people are watching recording. That's a good thing, because people are getting sending a screenshot, and they're asking the doubts. It's very good actually. And this will be the my class. And if any doubts, you can ask at any time. Okay, bye. Good night, everyone, and thank you for supporting through this 2-3 months journey from December, January, February. Like a lot of things we have discussed, and lot of we learned together. And I had to specially thank the students who attended live classes actually. And I think Satya is there, and Venkat or Jan. There are many students. They really uh, because I have, and all the students should thank them, because without interaction, because I can't discuss more, because without interaction I would have limited to my presentation only. But I gone behind the presentation because of the discussion only, the questions were raised in the live classes. And every student has to thank them also. And I personally thank them, and it's really very good interaction, at least for some students. And I hope to see you, like many of you in IRA as my juniors, and also how to, if I would have reached like ARS also there. I am also preparing, you also prepare well, and together we'll discuss more in the text, WhatsApp text or Telegram or group. Okay. In our live classes, I can't like live classes because my subscription is coming to end. Okay. Like I had to recharge for storage only. Then okay. Bye bye everyone. Thank you. Good night.
Thank you, sir. It was very nice discussing with you in the live classes, sir. It
Yeah,
cleared many doubts, sir. Cleared so many doubts.
yeah, thank you, thank you. Because of you
Okay.
and other students, really, I discussed more and we learned more. Actually. Even I also
Reading
read.
books won't clear that, sir. It only clears when we
Yeah.
discuss.
Yeah, really, really,
That level
actually,
of
because
understanding will come only after discussing.
yeah, and we'll remember for life actually if we discuss more. If you read the book, you'll forget everything. Okay,
Yes.
that's what I also learned many things actually because while teaching quantitative genetics also, the way I made the notes really, I took a lot of time to prepare these notes, but on that day actually no one was there actually. That's why I went very speed. Actually, every topic I thought it will go for a day. Like every topic is, I thought go for hour, but it finished in very early because nobody is there. Just I discussed, but
Yes, sir.
maybe
Actually, I went out, sir. I am
no,
not
no, 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 no. Like no, no,
unable
no.
to
I'm not
attend the
telling you.
live.
Yeah, yeah. Actually, if I at least you would have been there and actually training somebody as we were going train because at least you attended all the classes. Okay. And many of I think 9 to 10 students are attended very nice. And really I'm thankful to everyone. And those students were joined also because all together and I think I'm not accepting many new requests. I think many 10 to 15 students actually they're asked to join, but they because I denied to accept the course because most of them are PhD students. I don't want to them to be in the class, uh, and also I'm stopping the course because I can't able to manage the things because I had to have my research will start and that's why and even don't recommend any of your friends also because I'm not taking any more students. Okay, and this will be this whatever the numbers are there now, this will be the last batch. I think I hope this will be the last batch. If next year if any request come, then only I will give recording but I will not take live class. And really thank you everyone and once again thank you. Bye, good night. Night.
Thank you, sir. Good night.
Bye bye.
Nice.